Hi, my name is Dr. Brandi Kamermans, and welcome to my talk, Detecting and Identifying Saxitoxin-Producing Algae in the Salish Sea. I would first like to acknowledge our funding sources from the National Science Foundation and NIFA, as well as my colleagues at the Salish Sea Research Center, the University of Alaska Fairbanks, and Lummi Natural Resources. I am currently studying a marine ecosystem located on the Salish Sea. The Salish Sea borders both the United States and Canada and includes Vancouver and Seattle. The northern boundary of the Salish Sea is the Strait of Georgia, and the majority of the marine influence comes from the Strait of Juan de Fuca. I've added a white box to this map to designate where our study area is. Here is a close-up image of our field area. I have included the Strait of Georgia as well as the Fraser River to our north, and I've pointed out both Bellingham Bay and Lummi Bay and the city of Bellingham. We are currently located at Northwest Indian College, located on the Lummi Nation. The research I do is driven by the people that inhabit and have so for time immemorial the waters of the Salish Sea, the Lummi, or as they call themselves, the Laktamish. The Lummi people are the original inhabitants of Washington's northernmost coast in southern British Columbia. For thousands of years, they have worked, struggled, and celebrated life on the shores and waters of Puget Sound. I have added to this slide a map of Lummi Nation and indicating where Lummi Bay and Bellingham Bay are, as well as the city of Bellingham. I currently work at the Salish Sea Research Center, the mission of which is to prepare the next generation of environmental scientists and leaders. One of the key purposes of our research center is to enact data and food sovereignty and governance for the Lummi people. We are a team of researchers that utilize analytical chemistry, genetics, geosciences, and microscopy to provide both food and data sovereignty for the Lummi Nation. Lummi Nation has voiced a concern about local shellfish and the onset of harmful algal blooms, and we as a team of researchers are working towards data sovereignty and governance for the Lummi people and their subsistence harvesting efforts. Here I've added a schematic which first shows Alexandrium, a species of algae that produce a biotoxin called saxitoxin that accumulates in shellfish and crab. This is to depict how marine biotoxins are produced by aquatic microorganisms, or shown here, Alexandrium, and that these accumulate in shellfish. These toxins act as blockers of voltage-dependent channels, inhibiting the transmission of neuronal signals. The first symptoms would be tingling sensation in the lips, tongue, and throat, and numbness of the face, which may progress in the most severe and acute cases of intoxication to paralysis. At the SSRC, we study biotoxins of the saxitoxin group by collecting weekly samples at 3 marine sampling stations. We have spat data as well as cell counts that we provide to Sound Toxins. Sound Toxins is a diverse partnership of Washington state shellfish and finfish growers environmental learning centers, native tribes, and Puget Sound volunteers to keep track of harmful algal blooms in Washington state. I show again the image of where our data is collected in the marine settings of Lummi Bay and Bellingham Bay. This middle image is our spat bag that collects biotoxins on plastic beads. And on the far right here is an image of our tow net that we use to see what algae are in our marine waters using microscopy. In 2020, I was hired to design an assay to detect and quantify harmful algal blooms in both Lummi and Bellingham Bay. I first started this work by using SEM to image Alexandrium. That's the image I show here on the left side of the slide. I started experimenting with several different assays provided in the literature, and I got in contact with researchers using a commercially available assay through phytoxygen. Phytoxygen has an assay kit called DynoDetect that detects saxitoxin-producing genes from environmental samples. We have been purchasing the kit for about 6 months now and have data to show, some preliminary data to show that we are able to detect the saxitoxin-producing genes in Bellingham Bay. So on this slide, I'm showing the cartoon drawing of a dinoflagellate alongside a cartoon showing the ultrastructure of the dinoflagellates. And I've highlighted the nucleus. This is where the saxitoxin-producing gene is located. Saxitoxin biosynthesis is initiated by an enzyme. The enzyme contains 4 domains, and the 4th domain is highly conserved in several strains of saxitoxin-producing dinoflagellates. It is also absent from non-producing species. By utilizing the DynoDetect assay, we are able to collect environmental DNA from Bellingham Bay and conduct qPCR reactions. qPCR combines forward and reverse primers complementary to the gene of interest, and a TACMan probe that is also complementary to the gene of interest, and combines it with DNTPs and Taq polymerase. We are utilizing these reagents and a QuantStudio 6 thermocycler that is able to run 96 experiments at one time. We have nicknamed our thermocycler Tauntaun. Tauntaun is set up to run PCR at set PCR parameters that we specify. The first step is to denature our environmental DNA at 96 degrees. And at 61 degrees, the forward and reverse primers and our TACMan probe anneal to complementary regions of DNA that correspond to the saxitoxin-producing enzyme. As Taq polymerase begins to attach base pairs to the single-stranded DNA, it knocks off our hydrolyzing probe, including the fluorophore that is part of the hydrolyzing probe, and the fluorophore generates fluorescence that is then detected by Tauntaun. In this figure, you can see an epi tube that has no fluorescence and one that does have fluorescence, and it is this fluorescence that is detected by Tauntaun. Exponential curves that show the increase and then the plateau phase of fluorescence are shown here, and a threshold line or a CT value is crossed. And we compare these CT values among different samples to see the increase or the decrease of a gene of interest, and in this case it's the saxitoxin-producing enzyme. So returning to the circle of data sets that we are collecting, I'm going to combine our Hab cycling gene concentration data with microscopy data as well as biotoxin concentrations to better understand the bloom dynamics of saxitoxin-producing microorganisms in Bellingham Bay. I've shown here a map of Nauset Marsh system in Massachusetts and data that were collected in this ecosystem, because this is the goal for our study, is to combine cell counts with saxitoxin enzyme concentrations and toxin levels in Bellingham Bay, similar to how Murray et al. Have combined these data sets in a publication from 2019. Work is currently underway to utilize published saxitoxin-producing enzymes that have already been published and put into databases to generate our own qPCR assay. Our goal is to provide food and data sovereignty and governance for the Lummi Nation to better inform management of their marine resources. Thank you for your time, and if you have any questions, please feel free to contact me or ask me after this talk.